Introduction. Welcome to today's lecture on endocrinology and its role in lactation. In this comprehensive script, we will explore various hormonal factors that contribute to the intricate process of milk production and ejection. Understanding the complex interactions between hormones is crucial for medical professionals in the field of obstetrics and gynecology, as they play a critical role in supporting lactation and ensuring the well-being of both mother and infant. Now, let's dive into the fascinating world of endocrinology and lactation. Prolactin, a primary hormone secreted by the anterior pituitary gland, is instrumental in the initiation and maintenance of lactation. It stimulates the development of the mammary glands, promoting milk synthesis and secretion. Prolactin stimulates alveolar epithelial cell proliferation, increasing the number of milk-producing cells in the breast. Additionally, it binds to prolactin receptors on the mammary epithelial cells, activating signaling cascades that lead to milk protein synthesis. Furthermore, prolactin inhibits the release of gonadotropin-releasing hormone, GnRH, from the hypothalamus, suppressing ovarian follicular development and thus preventing ovulation, which facilitates the extended periods of lactational amenorrhea often observed in lactating women. Oxytocin, released from the posterior pituitary gland, plays a pivotal role in the milk ejection reflex, also known as the letdown reflex. During breastfeeding, suckling stimulates mechanoreceptors in the nipple, triggering oxytocin release. Oxytocin acts on myoepithelial cells surrounding the alveoli and ducts, causing their contraction and ultimately resulting in the expulsion of milk into the lactiferous sinuses. The release of oxytocin is regulated by a positive feedback loop. As the infant suckles, oxytocin release is enhanced, leading to increased milk ejection. This feedback mechanism is essential for efficient milk removal and maintaining an adequate milk supply. Milk production, also known as lactogenesis, is regulated by a delicate interplay of hormones. Initially, during late pregnancy, high levels of estrogen and progesterone inhibit milk synthesis. However, following delivery, the abrupt decline in these hormones allows for the initiation of lactation. Once the feedback inhibition from estrogen and progesterone is removed, prolactin dominates and stimulates the synthesis of milk components in the mammary glands. Additionally, lactogenesis requires the coordinated actions of cortisol, insulin, thyroid hormones, and growth hormone, as we will explore in subsequent topics. Estrogen and progesterone, produced by the ovaries and placenta during pregnancy, are essential for the development and maintenance of the mammary glands. These hormones promote epithelial cell proliferation and the differentiation of alveolar cells in preparation for milk production. However, once lactation is established, high levels of estrogen and progesterone inhibit milk synthesis by exerting negative feedback on the hypothalamus and anterior pituitary, thereby reducing prolactin release. Thyroid hormones, particularly thyroxine, T4, also contribute to the regulation of lactation. They enhance the synthesis and secretion of prolactin and stimulate the growth and differentiation of mammary epithelial cells. Hypothyroidism, characterized by insufficient thyroid hormone production, can lead to lactation failure and decreased milk production. Glucocorticoids, such as cortisol, have both direct and indirect effects on lactation. They support the differentiation of mammary epithelial cells, ensuring proper glandular development. Glucocorticoids also regulate the expression of genes involved in milk protein synthesis. However, excessive levels of glucocorticoids can inhibit lactation, emphasizing the importance of maintaining a balanced hormonal milieu. Growth hormone, GH, influences the growth and development of the mammary glands. It promotes the elongation and branching of ductal structures within the breast, facilitating milk production. GH also stimulates the production of insulin like growth factor 1, IGF-1, in the liver, which enhances milk protein synthesis. Disruptions in the GH signaling pathway can lead to impaired mammary gland development and consequently affect milk production. Insulin, primarily known for its role in glucose metabolism, also exerts effects on lactation. It promotes milk secretion by facilitating glucose and amino acid uptake in the mammary glands, providing the necessary energy and substrates for milk synthesis. Insulin acts synergistically with other hormones, such as prolactin and glucocorticoids, stimulating milk production and ensuring optimal lactation. Leptin, an adipose tissue-derived hormone, influences lactation by modulating appetite and energy balance. It acts on the hypothalamus to suppress appetite and decrease food intake, aiming to maintain optimal energy levels required for milk production. Leptin also regulates lactation indirectly by influencing the responsiveness of the mammary glands to prolactin. Prolactin-releasing peptide, prrp, is a neuropeptide that stimulates prolactin release from the anterior pituitary gland. It acts directly on lactotroph cells, enhancing prolactin synthesis and secretion. prrp thus plays a crucial role in maintaining high prolactin levels necessary for sustained lactation. Dopamine acts as a tonic inhibitor of prolactin secretion. Dopaminergic neurons in the hypothalamus release dopamine into the pituitary portal circulation, where it binds to D2 receptors on lactotrophs, preventing the secretion of prolactin. However, during breastfeeding, the suckling stimulus inhibits dopamine release, consequently alleviating the inhibitory effect on prolactin secretion and facilitating lactation. Stress, both physiological and psychological, can negatively impact lactation. Stress hormones, such as cortisol and catecholamines, suppress the release of prolactin, thereby inhibiting milk production. Stress can also disrupt the oxytocin-mediated milk ejection reflex, leading to inadequate milk removal. Providing ample support and reducing stressors are crucial in maintaining optimal lactation by ensuring proper hormone release and milk ejection. In conclusion, the regulation of lactation involves an intricate interplay of various hormones, each with specific roles in mammary gland development, milk production, and milk ejection. Prolactin, oxytocin, estrogen, progesterone, thyroid hormones, glucocorticoids, growth hormone, insulin, leptin, prrp, and dopamine all contribute to the complex process of lactation. Understanding the hormonal control of lactation is essential for healthcare professionals, enabling them to support breastfeeding mothers and address potential challenges that may arise. By comprehending the intricate dance of hormones, healthcare providers can optimize lactation outcomes, promoting the health and well-being of both mothers and infants. Thank you for joining today's lecture, and I look forward to our next session where we will delve into another fascinating topic in endocrinology.